Hi everyone, welcome back to discuss further formulation development stage 4, which is part 4. We have discussed a lot of factors in previous sessions. In this model, we'll speak about certain important process characterizations which has to be considered as a part of developmental activities. We will take one example, any of the dosage form, and list down the tests are required, studies are required to understand the robustness of the formulation. Process characterization study is a method to identify and qualify the significant source of variations in the manufacturing process, storage, or stability, which will impact the safety and efficacy of the drug product. For example, if you see tablets and capsules, we have to understand a different variability. Maybe you can understand the granulation time, the holding time of the granulation fluids, the holding time of the granuled powder, the holding time of your semi-finished goods, or the compatibility of the solutions which is being used with whatever the components, either it is a plastic or it is a glass or it is a stainless steel. Each and every compatibility have to be generated as a part of process characterization study. In this case, we have presented considering if you are developing a parenteral dosage form, what all the studies has to be performed. You have to do a drug product solutions with a manufacturing vessel, either it's a glass, a plastic bag, or a stainless steel. And of course, there is an exposure of filter membrane, which is PVDF, PVC, nylon. There are multiple filter membranes are available. You have to select these filter membranes and perform a compatibility studies and ensure that you will do a proper validations for the respective filter. And also tubing, freezer, temperature excursions, and photo stability. We can discuss one by one how these studies are going to be performed, how these studies are important to your process characterization and the whole product development. Manufacturing, why it is important? Because every manufacturing vessel has certain kind of abrasions and wear and tear, or certain kind of elements may get interact with a drug product. Again, the drug product, it may be acidic in nature or in alkaline in nature or in the neutral form. Accordingly, the drug product solutions will interact with the manufacturing vessel, whether it's a SS or it's a glass or LDP bag, because nowadays the manufacturing happening by using LDP bag also. So where it is necessary, a formulation scientist should understand the each components, how it has been made, what kind of vessels needs to be used to manufacture your final drug product. Hence, it is recommended to study the whole time in a selected vessel before you proceed for any kind of manufacturing at your manufacturing site. The next study is a filter compatibility study. The filter compatibility study, basically, it has to be done for every solutions, whether it is injectable or it is ophthalmic or it is nasal or any oral solutions. So this is a very, very important study because there are certain extracts or certain kind of filter residues may get into your product while filtering the solutions, because the filtration happens with either by the vacuum or by the pressure generated using either nitrogen or the compressed air. There are multiple chances where your filter membranes may get extracted by your drug product solutions. So it is important for us to study these filter membranes also while performing a developmental study for any kind of solutions. Here we have presented here few filter membranes where hydrophilic and hydrophobic membranes of any vendor. Maybe you can select it, polyvinylidene fluoride or polyether sulfone or nylon or cellulose. There are multiple filters are available. Using this, you can perform this study either by soaking a solutions into it or by passing the solutions and collect a predetermined interval. The samples shall be collected the predetermined interval. The sample shall be tested for your critical quality attributes and see is there any kind of difference in terms of your test results in comparison with your control. If there is, then you may have to relook at your filter membranes and followed by the filter validation has to be done where you are showing that your product need to be aseptically filtered. It is applicable for injectable and as well as ophthalmic solutions. Here, where we have just presented here how the static filter membrane compatibility has to be done, what are the tests are important. You can perform drug content, related substance, of course preservative, because preservative have a tendency to adsorb on any kind of filter membrane or tubings, any plastic surfaces. It is important to us to study the adsorption capacity of preservative on membranes. Coming to tubing compatibility study, because tubing is being used to transfer a solution from manufacturing tank to the filling area by the filtration process, then the filling area to your filling line, then filling line to your manifold and manifold to your vial or ampule, whatever the packaging materials are present in your line. So this is very important because most of the tubes are incompatible with a drug product solution, particularly on pharmaceutical drug product solution, because it has a buffers, it has certain kind of preservatives, and it has certain kind of organic solvents. With respect to drug product, the compatibility may get disturbed because all the tubes are not compatible with your drug product solutions. During your development, a formulation scientist should study at least 3 to 4 tubes to ensure that you are not absorbing any kind of impurities or any kind of leachables or any kind of extractables. You can also check from a vendor how this tubing need to be selected, what are the characteristics are required to study for your product, because the manufacturer also perform a tubing extractable study that would help you to understand how this tubing has been characterized at their facility. You can look at the data of physicochemical test, and certain people have done a validations on their elemental impurities, and extractables would be done for specifically non-volatile residues, pH, turbidity, and certain leachable components also. They would characterize and list down what are the possible components get leaches from the tubing based on the certain solvents. The data would be very helpful for the formulation scientists when they perform the study. So similarly, in this case also you can do a whole time study with your tubing with a predetermined time point and collect the samples and test based on your critical quality attributes. And also it is highly recommended to perform a leachable for the test tubing, transfer tubing also. Coming to a freeze thaw or a thermal cycling study. This is a study should be done for any liquid injections where you need to understand when we transfer a product from your distribution center to across the global, because when you transfer it there will be a shipping condition changes and there will be a lot of temperature variables. It could be a freezing condition or it could be hot and humid conditions. To cover this, we perform these studies using our samples with an exorbitant conditions. Maybe a two extreme conditions shall be selected. If your product is stable at long term as at 25 degrees Celsius, you can choose a freezing conditions and as well as an accelerated conditions of 40-75 and thaw the samples for 2 days in each storage conditions with the 3 cycles. And those samples also shall be tested and understand will our samples producing any kind of changes on shipping the samples from one place to other place or one condition to other conditions. So this would give a confidence for us when we transfer the product or shipping the product from your distribution center to various countries, because there is a difference in the temperatures and the humidity across the globe. Here are the studies. We have just tabulated for your better understanding. So this is something like you can get it referred from. There are a lot of discussions and guidelines are there. You can refer it to PDA, which would give you a lot of informations. From there we have captured over here for your understanding. Coming to temperature excursion, this is also a similar kind of a study where you can also see that a temperature excursion study defined in the WHO model guidance, an excursions event in which a time temperature sensitive pharmaceutical product is exposed to temperatures outside the ranges prescribed for storage and transport. The temperature ranges for storage and transport may be same or different. They are determined by the product manufacturer based on stability data. There is a case study here we have presented. Neostigmine methyl sulfate injection should be stored at 20 to 25 degrees Celsius, but the excursions permitted to 15 to 30 degrees Celsius. So the temperature excursion study should be conducted at minus 20 degrees Celsius for a week and 60 degrees Celsius for a couple of days. Again, this is also is referred in a PD, one of the PDA guideline. This gives you to study your excursions of any of the product, because many products we can say you can store the product at 25 degrees Celsius and then you can provide certain excursions, how long you can permit your product beyond your storage conditions. Maybe you can store the product for 30 degrees Celsius for 15 days or 7 days. Such kind of excursion shall be provided based on this study. So here we are. So test should be performed whenever you do this temperature excursion study. You can perform the assay, related substance, if any preservatives, pH and color and clarity. If at all, if you wanted to perform a particulate matter also would help you if any changes in the clarity of the solution. So the label describes here what is an excursions have been provided for this particular product. Coming to photostability. Photostability is very important study has to be done with all stages, either in the development or it is in a technology transfer. There is a defined guidelines for a drug substance and drug product in ICH Q1B. A study has to be conducted for drug substance are directly exposed to sample in petri dish and your immediate container along with a market pack. All these samples has to be studied against a control sample which is wrapped with aluminum foil. The samples shall be tested once you complete the study based on ICH Q&B recommendation. The samples should be tested for all the critical parameters and you can compare and summarize the results. Either your product is stable at stable photostable conditions. If not, there would be a recommendation has to be made. If the immediate container shows certain kind of reactions with the photo, then you may have to give a recommendations, keep the product into the secondary pack. If your immediate content does not show, then it is well and good. You don't have to give any major recommendations because your product can be stored at any storage conditions. So here are the study plan. We have just made one smart artwork, and basically the samples have to be kept in the photostability chamber, and it has to be exposed not less than 1.2 million lux hours and near ultraviolet not less than 200 watts per hour per square meter, and followed by the samples should be tested for the chemical. And next, a manufacturing process optimization. Why it has to be done? Because every manufacturing condition has to be optimized. Each manufacturing process has to be optimized. If you are preparing a granulation, the granulation time, your addition time, and if you are making a kind of a top spray granulations, you have to control your blower speed, you have to control your spraying time. Everything has to be optimized. This needs a design of experimentation process where you can provide your design space. What is your operating range for each process? If you are making a solutions, you should know the stirring time, you should know the RPM, and you should know the sequence of additions, and you should know the excipients range shall be used to get your desired product quality. Every product, the manufacturing process has to be optimized. If there is no optimization has been done, then you will be ending up with a lot of challenges when you transfer a product. Recent days, a lyophilization process also is being questioned by an FDA to optimize the process and to create a design space. Hence, we recommend you to study and design a manufacturing process optimization design space. So these are the guidances I would recommend you guys have to go through before you go for any kind of interviews or any kind of discussion with any pharmaceutical industries. Of course, this is not limited to. You can refer as much as possible, but these are the very, very essential guidance which we would like to recommend for your future. Thank you all and wish you good luck.